Hi, I'm David Woolard, biology professor at Chattanooga State. Welcome to this tutorial on CRISPR. It's very possible that you've already heard of CRISPR. It's been in the news quite a bit lately. If you haven't heard of it, then it's definitely time that you did. Already hailed as the greatest discovery of the century, CRISPR is a powerful biotechnology tool giving scientists unprecedented access to the genetic makeup of all living organisms, including humans. It originally evolved as an adaptive immune system in bacteria to defend against viruses. But when artificially harnessed in the laboratory, it allows scientists to accurately and precisely edit genes, almost as if using a word processor. Using CRISPR technology, scientists can correct genes, delete genes, and introduce new genes. The potential applications are mind-boggling, but they also open an enormous range of bioethical questions regarding how and when the technology should be used. Some have cautioned that CRISPR will move us beyond traditional boundaries of science, to the point of playing God. We'll explore some of these applications later, but first let's see how CRISPR was discovered. The story of CRISPR begins in 1987 when Japanese biologist Yoshizumi Ishino was studying DNA in the common gut bacteria E. coli. While sequencing part of the E. coli chromosome, he discovered several short repetitive sequences separated by short random spacer sequences, approximately 20 nucleotides long. These short repeat sequences would come to be called CRISPR, clustered, regularly interspaced, short palindromic repeats. Ishino's paper famously concludes, "The biological significance of these sequences is not known." CRISPR repeats were soon discovered in many other types of bacteria. The sequences of the repeats were consistent and predictable, but the 20 nucleotide spacer sequences always appeared to be random. That is, until 2005 when Francisco Mojica, working in Spain, discovered that the spacer sequences were identical to sequences of bacteriophage DNA. Bacteriophage are viruses that infect bacteria cells. It seemed that bacteria were storing fragments of this viral DNA in between the CRISPR repeats. Mojica speculated that the CRISPR system might represent some type of bacterial defense against the viruses, an idea that would later be proven correct. The proof came from the research of Rudolf Barrangou in the US and Philippe Horvat in France, two microbiologists working for Danisco. Danisco is a major producer of yogurt, which of course requires cultured bacteria, including the species Streptococcus thermophilus. Infection of bacteria by bacteriophage presents a major problem in the yogurt industry, so the Danisco scientists were very interested in the possibility of a bacterial immune system. They performed a fascinating and enlightening experiment. First, they sequenced the naturally occurring wild strain of Streptococcus thermophilus and, as expected, found CRISPR sequences within the genome. They then incubated the bacteria in the presence of a bacteriophage virus known to infect the cells. They then transferred the culture to agar growth plates and incubated the plates in hopes of finding mutant colonies resistant or immune to the virus. Sure enough, there were 9 mutant colonies. They sequenced the mutant genomes and, much to their delight, identified a new CRISPR sequence in the genome, matching that of the bacteriophage. Clearly, CRISPR was some type of adaptive immune system. But how does the system work? The next piece of the puzzle came from Stan Browns while working in the lab of John van der Oos in the Netherlands. Browns discovered that bacteria transcribe the CRISPR region of the genome into a single large RNA molecule. This RNA molecule is then separated into smaller RNA segments called CRISPR RNA. Each CRISPR RNA contains a CRISPR repeat along with a short viral DNA spacer sequence. biologists began to suspect that the CRISPR RNAs might provide immunity by acting as interference RNAs, a phenomenon found in some eukaryotic cells. Spoiler alert, the CRISPR defense is not based on interference RNA, but it was this intriguing possibility that caught the interest of Cal Berkeley biologist Jennifer Doudna. Doudna was an RNA expert and couldn't resist looking into the CRISPR system. Doudna suspected that a role might be played by some genes located near the CRISPR region. These CRISPR-associated genes, or Cas genes, encode protein enzymes and had been discovered by Ruud Janssen back in 2002. Doudna's lab was able to work out the roles of two of the enzymes. Cas1 was responsible for splicing the viral DNA fragments into the CRISPR region of the chromosome, while Cas6 cut the transcribed RNA into the smaller CRISPR RNA segments. But still, no explanation of how the entire system worked. That would all change, however, following a chance encounter between Doudna and another biologist, Emmanuel Charpentier, at a conference in Puerto Rico in 2011. Charpentier was studying yet another Cas enzyme called Cas9, which had been found in Streptococcus pyogenes, the bacteria responsible for causing strep throat. Cas9 is a fascinating protein with the intriguing ability to cut DNA molecules like a pair of scissors. Another class of DNA-cutting enzymes, called restriction enzymes, had been known for some time. But restriction enzymes always cut DNA at a short, specific, predefined sequence. Cas9, on the other hand, has the potential to cut DNA at any particular sequence. Indeed, the enzyme must be told where to cut the DNA. Interestingly, these instructions come from an RNA molecule that attaches itself to the Cas9 enzyme. In 2012, Emmanuel Charpentier and Jennifer Doudna were able to put all of the pieces together and demonstrate how the CRISPR system operates in bacteria to serve as an adaptive immune system against viruses. When a virus infects a bacteria cell, it first injects its viral DNA into the cell. The viral DNA instructs the bacteria on how to make many more copies of the virus, and the bacteria cell eventually ruptures like a piñata from the production of new viruses. However, if the bacteria somehow survives the infection, it will take a fragment of the viral DNA and splice it into its own chromosome. It then adds CRISPR sequences on either side. In fact, whenever a bacteria cell survives a viral infection, it will perform this task. The bacteria is creating a database of known viruses, much like an FBI Most Wanted list. The bacteria is keeping mugshots and fingerprints of viruses it has previously fought in the form of short DNA sequences. Interestingly, this Most Wanted list gets passed on genetically to all descendant cells of the bacteria, so that subsequent generations will recognize the viruses even before being attacked. But how does the CRISPR system actually defend against the viruses? As mentioned previously, bacteria transcribe the entire CRISPR region into complementary RNA, which is then cleaved into separate pieces of CRISPR RNA. Recall that each CRISPR RNA contains the CRISPR repeat, along with a short 20 nucleotide stretch of viral DNA. The CRISPR RNA is then loaded into the Cas9 enzyme by anchoring it to a piece of tracker RNA. Charpentier had already discovered tracker RNA in conjunction with Cas9, but had not been able to determine its function. It was now found to serve as a universal attachment site for CRISPR RNAs. Thus, the Cas9 enzyme can be programmed by the cell to cut at whichever target sequence is specified at the end of the CRISPR RNA. The cell is now ready and waiting should that targeted strain of virus attempt to enter the bacteria. If viral DNA does enter the bacteria cell, the Cas9 CRISPR RNA complex will recognize the DNA by complementary base pairing, open it up, and cut it. Once cut, the viral DNA is no longer active, nor able to harm the cell. It's a truly fascinating system, with the ability to learn and adapt. Bacteria are always updating their database of viral fragments. It's not unlike our own immune system, which learns to recognize harmful germs throughout our life and produces highly specific antibodies to fight them off. With the CRISPR mechanism solved, Doudna immediately wondered if it might be possible to customize the CRISPR system in order to cut a DNA sequence of one's choosing. Could researchers create their own CRISPR RNA and load it into the Cas9 enzyme? The first step was to simplify the CRISPR RNA tracker RNA complex. Doudna and Charpentier essentially fused the two RNAs together to produce a single, what they called, guide RNA. The next step was to synthesize a guide RNA with a specific target sequence of their choosing, to see if they could actually program the Cas9 enzyme. Doudna worked with her postdoc, Martin Yennick, to perform the crucial experiment. They decided to target the green fluorescent protein, or GFP gene. Although the gene naturally occurs in jellyfish, it can be easily introduced into E. coli bacteria. When present, bacteria express the gene, which encodes a green fluorescent protein. This causes the bacteria to appear green when grown in culture. Doudna and Yennick wanted to see if they could cut the gene using a customized Cas9 guide RNA complex. Here's the E. coli DNA with a copy of the GFP gene. The gene contains a specific cutting site for the SAL1 restriction enzyme near the start of the gene. Thus, this region of the DNA could easily be cut using SAL1, just as researchers have been doing for decades. Doudna and Yennick chose a unique sequence further downstream in the gene and designed a guide RNA to recognize that sequence. Their assumption was that if the GFP gene was cut by both the SAL1 restriction enzyme and their customized Cas9 guide RNA complex, it would produce a DNA fragment of a known length that could be visualized using gel electrophoresis. To confirm their approach, they designed guide RNAs targeting additional sequences in the GFP gene so as to produce additional fragments of known lengths. The experiments were a resounding success. Here is a picture of their resulting gel. As you can see, DNA fragments of each expected length were produced. CRISPR could quite easily be customized to cut DNA at any sequence the researcher chose. Fast, accurate, inexpensive genome editing had arrived almost out of nowhere. Doudna and Charpentier published their work on CRISPR in June of 2012. In yet another understatement in the history of CRISPR, we propose an alternative gene editing methodology based on RNA-programmed Cas9 that could offer considerable potential for gene targeting and genome editing applications. Ironically, another scientist in Lithuania, Virginijus Siksnis, had also worked out the functioning of CRISPR-Cas9 around the same time. But publication of his research was delayed by extensive peer review. Unfortunately, as a result of the delay, Siksnis's work is often overlooked. Once the CRISPR-Cas9 system was worked out in bacteria, Doudna immediately wondered if a similar system was present in the more complex eukaryotic cells of animals. The answer is no. However, she did find that one can artificially introduce the CRISPR-Cas9 system into eukaryotic cells, and the system works just fine. Indeed, that's exactly what she and Yennick did. They designed and deployed a customized CRISPR-Cas9 system to target and disable the clathrin light chain A gene in human embryonic kidney cells. Obviously, the implications for new types of genetic research were profound. They published their work in January of 2013. But here's where the story gets really interesting. Around the same time, Feng Zhang at MIT and George Church at Harvard published similar studies involving the use of CRISPR to edit human cells. Zhang and Church, however, took the method one step further. It turns out that when DNA is cut in a eukaryotic cell, the cell will immediately try to repair the break. Recall that human cells do not naturally have a CRISPR system, so they perceive any broken DNA as a problem that needs to be fixed. As such, the cell will attempt to repair the DNA using one of two repair options. The first option is called non-homologous enjoining, in which the cell will simply try to glue the broken DNA molecule back together. It does this by inserting a few random nucleotides to serve as molecular glue. If the cut sequence of DNA was part of a gene, however, then this repair mechanism introduces mutations into the gene, and the gene will no longer function. Thus, the gene is silenced or knocked out. This is what Doudna and Untitled had accomplished by targeting the CLTA gene in kidney cells. The second option, called homology-directed repair, is quite different. If a piece of DNA is available with sequences that match the broken ends of the cut DNA, then this homologous DNA can serve as a template for repairing the broken DNA. Hence the name homology-directed repair. In diploid eukaryotic cells, this role is served by the homologous chromosome. However, if donor DNA molecules are introduced into the system with ends matching the tips of the broken DNA, then the donor DNA can play the role of template molecule and direct the repair process. Furthermore, if the donor DNA molecule happens to include a gene, then this gene will be incorporated into the DNA during the repair process. Church and Zhang use this latter method, homology-directed repair, to incorporate the green fluorescent protein gene into cultured human embryonic kidney cells. On the left, you can see human cells without the GFP gene. On the right are cells with the GFP gene introduced. Thus, not only can CRISPR be used to turn existing genes off, referred to as a gene knockout, CRISPR can also be used to incorporate or knock in new genes. As such, the prospect of using CRISPR to treat human genetic diseases became a very real possibility. Consider cystic fibrosis, for example. Cystic fibrosis is characterized by an excessive accumulation of mucus, particularly in the respiratory tract. The mucus clogs up the respiratory tubing, making it extremely difficult to breathe. The problem ultimately stems from a genetic error. The CFTR gene, which normally encodes a protein used to transport ions across the cell membrane, is faulty. As such, the correct protein is not made, the ions are not transported, and the result is an accumulation of thick, heavy mucus. CRISPR could potentially be used to correct this genetic mistake. Scientists could create a guide RNA molecule targeting the faulty CFTR gene. The guide RNA would then direct Cas9 to the faulty gene, allowing Cas9 to cut the gene at the site of the error. This alone would not solve the problem, as the gene was already not functioning correctly. But if a piece of donor DNA containing the correct version of the CFTR gene were also provided, then this new correct version would be spliced into the chromosome in place of the faulty gene. This is exactly what Hans Clevers did in 2013. He used CRISPR to correct the cystic fibrosis mutation in human cells cultured in the laboratory. Although Clever's experiment was groundbreaking, there is one obvious problem. Every cell in an adult cystic fibrosis patient possesses the faulty CFTR gene. As such, scientists would have to find a way to introduce the CRISPR repair mechanism into every adult cell, or at least into those of the respiratory tract. This would be extremely difficult to do. The ideal scenario would be to introduce CRISPR-Cas9 into the fertilized egg or early embryonic stage of someone with cystic fibrosis, so that all future body cells of the adult organism would contain the corrected gene. Many scientists have expressed concern, however, with performing such an experiment. It's one thing to treat the existing cells of an adult. It's quite another to genetically alter a human embryo, as this opens up additional ethical questions. CRISPR was opening a Pandora's box of bioethical questions. In January of 2015, Jennifer Doudna organized an international meeting of scientists and policymakers to discuss the pros and cons of the potential uses of CRISPR, including the editing of human embryos. The attendees agreed that editing human embryos crossed a bioethical line and began authoring a self-imposed ban on such a procedure. Little did they know that a Chinese scientist named Junjiu Huang had already crossed that line. Junjiu Huang had performed the first CRISPR editing of human embryos. His goal was to correct a gene associated with a blood disorder called beta thalassemia. Huang injected 86 embryos with CRISPR-Cas9. Only 4 embryos, however, were edited successfully, and many were found to have unintended or off-target mutations. The experiment was so controversial and arguably unsuccessful that Huang's paper was rejected by both Science and Nature, the world's premier scientific journals. In 2016, Chinese scientist Lu Yu reported the first use of CRISPR to treat an adult patient. The patient had a type of lung cancer. Unfortunately, the patient's T cells, a type of white blood cell, were unable to recognize and destroy the cancer cells. Lu removed the patient's T cells and then used CRISPR to disable the PD-1 gene. This alteration would allow the T cells to attack the cancer cells. The T cells were then injected back into the patient with the hope that they would fight off the tumor. Time will tell if the experimental treatment is successful, but it is clear that science is already moving forward with CRISPR-based therapies. The groundbreaking research of Chinese scientists has initiated a science war between the United States and China. It's a battle not unlike the space race between the United States and Russia during the 1960s. China has less stringent regulations regarding biomedical research, including the use of human embryos, so their scientists are able to push the boundaries of research further. Sensing that such experiments were ultimately inevitable, U.S. and European scientists have loosened their self-imposed regulations and have begun to explore similar types of experiments. In fact, Shukrat Mitalapov, working at the Oregon Health Sciences Research Center, became the first U.S. scientist to perform CRISPR editing of viable human embryos. Mitalapov used CRISPR to edit a gene associated with excessive thickening of heart muscle. Although successfully edited, the embryos were not implanted for further development. The potential biomedical applications of CRISPR are far-reaching. Thousands of individuals die each year in need of an organ transplant – heart, lung, kidney, liver, etc. Patients must wait until an appropriate donor is available, often the result of a tragic accident. Even then, the donated organ must be a very close genetic match to ensure that the organ is not rejected. Jun Wu, a research scientist at the Salk Institute in California, is pioneering a solution. Wu's goal is to develop interspecies organ donation. As part of his research, he and his colleagues used CRISPR to grow rat organs inside the body of a mouse. First, they used CRISPR to turn off the organ-producing genes inside a mouse blastocyst. They then inserted rat stem cells into the mouse blastocyst in hopes that the rat cells would generate the missing organs. The experiment worked, resulting in a hybrid organism called a chimera. The organism on the far left is 100% mouse. The organism on the far right is 100% rat. The second organism is a mouse-rat chimera created by inserting rat cells into a mouse blastocyst. The third organism was created by placing mouse cells into a rat blastocyst. Wu and his colleagues managed to produce a mouse with rat organs, heart, eyes, pancreas, etc. These organs could be harvested from the chimeric mouse and donated to a normal rat without risk of rejection. Wu would like to use this technology to produce human organs inside another species of animal. Obviously, rats and mice are too small, but it turns out that pig organs are remarkably similar in size and structure to human organs. Scientists have long wondered if it would be possible to genetically alter pig tissues so as to make them suitable for human transplant. CRISPR finally offers a means to do it. Using the same approach, Wu inserted human stem cells into a pig blastocyst, and the experiment worked. You're looking at a picture of the first pig-human embryo, a chimera. The embryo was destroyed before being allowed to fully develop. It is worth noting that Wu's research, although legal, was funded by private foundations, as government funding was not an option. There are many other CRISPR applications currently under development, with billions of dollars at stake in terms of patents and prizes. Recall that Feng Zhang was one of the first to use CRISPR on human cells. His success instigated an intense patent dispute between his research team at the Broad Institute of MIT and Harvard and the team of Doudna and Charpentier at the University of California at Berkeley. Although Doudna and Charpentier first published the CRISPR-Cas9 technology, the Broad Institute argued that Zhang modified the procedure in significant ways to exploit homology-directed repair. The Broad Institute also paid a fee to have their patent application expedited through the process. The courts ruled in Zhang's favor in February of 2017, a decision with enormous financial implications. Doudna and Cal Berkeley appealed the decision. The potential applications of CRISPR are limitless. Scientists have already used CRISPR to treat diabetes, muscular dystrophy, and blindness in mice. CRISPR has been used to make cultured cells immune to the HIV virus. In addition to biomedical applications, CRISPR has the potential to revolutionize the food industry. The United States Department of Agriculture has already approved the first CRISPR-edited food item, white button mushrooms. Scientists at Penn State University used CRISPR to disable an enzyme that normally causes the mushrooms to brown, thereby extending their shelf life. Interestingly, in March of 2018, the USDA noted a distinction between gene modification and gene editing, at least with regards to plants. Gene editing refers to changes that, theoretically, could be produced and propagated naturally through mutation and selective breeding, even if the changes are actually accomplished using biotechnology tools, such as CRISPR. As such, many CRISPR-edited foods will not be subject to regulations, nor labeled as genetically modified organisms, GMOs. For example, scientists simply disabled an existing gene that caused the mushrooms to brown, an event that theoretically could occur naturally. Thus, the mushrooms are not subject to regulation nor considered GMOs. CRISPR can be used to knock out genes, but it can also be used to edit promoter sequences, thus introducing a type of volume knob control over genetic expression. Again, this would simply be gene editing and not gene modification. Fungus-resistant produce and reduced gluten wheat, along with many other edited food items, will be available in the near future. Decisions regarding livestock have yet to be made. Consider the following example. Here is a picture of a genetically modified salmon. Scientists added a growth hormone gene from another species of salmon, as well as a regulatory sequence from yet another fish species, to ensure that the growth hormone is constitutively expressed. The result is a GMO salmon that grows 5 times faster and 5 times larger than the wild-type salmon. Approval of the salmon took over 20 years at a cost of $60 million. Nonetheless, a New York Times survey found that 75% of consumers would not eat the GMO fish. Now consider the Belgian blue breed of cattle. Belgian blues have an abundance of lean muscle due to a natural mutation in the myostatin gene. The myostatin protein normally exerts negative feedback on muscle growth. A mutation knocking out the myostatin gene occurred many years ago, resulting in this fascinating breed. This organism is not a GMO because the mutation occurred through natural events. Scientists, however, are now using CRISPR to intentionally knock out the myostatin gene in a variety of livestock, hoping to produce cows, pigs, goats, and other animals with exaggerated amounts of lean muscle mass. Since the myostatin mutation could theoretically occur naturally and be propagated through selective breeding, meat from such livestock may not necessarily be labeled as GMO. Yet another fascinating application of CRISPR is a tool called a gene drive. Originally conceived by Harvard biologist Kevin Esvelt, a gene drive is a synthetic segment of DNA that includes the Cas9 gene and a guide RNA gene, along with a particular gene of interest, also called payload DNA, all in one self-functioning unit. The payload DNA might be a new gene or a modified version of an existing gene. Once a gene drive is introduced into the chromosome of a diploid organism, the drive will generate a Cas9 guide RNA complex that will cut the homologous chromosome and copy the original gene drive into the cut chromosome via homology-directed repair. With the gene drive now present in both chromosomes, the organism is homozygous for the drive, including the payload DNA. Gene drives are capable of knocking in or knocking out genes in an organism. More importantly, once incorporated into an organism, the gene drive can be propagated via sexual reproduction into entire populations of organisms, thus allowing for genetic modification at the population level. Consider the following diagram showing how a gene is propagated in a population via normal inheritance, that is, with no gene drives involved. If one of the original parents is heterozygous for the gene of interest, then it should statistically be transmitted to half of the offspring. This pattern of inheritance would continue in subsequent generations, with the gene never accumulating to an appreciable level within the population. Now, look at how the gene could be propagated using a gene drive. The original parent would be homozygous for the drive, including the gene of interest. Zygotes in the first generation of offspring would initially be heterozygous for the gene drive, but the drive would copy itself along with the payload DNA into the other chromosome, thus ensuring that the offspring are homozygous. This pattern of inheritance would continue in subsequent generations, with the gene drive and payload gene becoming increasingly present within the entire population. So how might this technology be used? In one interesting application, scientists are hoping to use gene drives to eliminate malaria, a disease that continues to kill over 1 million people every year. The malarial parasite is transmitted by the Anopheles mosquito. In an attempt to address the problem, Ethan Beer's lab used CRISPR to engineer mosquitoes genetically incapable of transmitting the malarial parasite. If introduced into the wild population, the gene drive could spread and drastically reduce transmission of the parasite. Austin Burt's lab went another direction with gene drives and engineered the mosquito so as to make females of the species reproductively sterile. This second approach could conceivably drive the mosquito species to extinction. Gene drives represent an extremely powerful technology, with the potential to alter entire populations of organisms. The enormous power of gene drives has not gone unnoticed by the United States government. In 2016, the Director of National Intelligence added gene editing to a list of threats posed by weapons of mass destruction and proliferation. Indeed, Kevin Esvelt's lab is already working on an antidote to gene drives, specifically a gene drive program to remove another gene drive. Perhaps most controversial, CRISPR editing could easily be performed on human sperm and egg cells prior to in vitro fertilization. In this way, parents could correct genetic problems even before conception. Of course, they could also customize many other traits, true designer babies. Scientists, however, have traditionally been reluctant to allow editing of human germline cells, as the edits would be permanently passed on to future generations without consent and without full knowledge of long-term consequences. Pressured by the advances being made in China, the National Academies of Science recently updated their position on gene editing of germ cells. In an abrupt and significant change, they now support gene editing of both embryos and germ cells in cases of serious disease. The National Academies of Science, however, cannot enact laws. Rather, they provide professional advice to the government. Ultimately, it is the President and Legislature of the United States that can establish laws and regulations on such matters. In 1859, Charles Darwin published his renowned theory of evolution by means of natural selection. His theoretical recipe required 3 ingredients: 1. Natural occurring variation, a seemingly random component; 2. Selective preservation of those variations that confer reproductive success; and 3. Long periods of time for the first 2 ingredients, chance and necessity, to play themselves out. For all intents and purposes, nature has been in charge, incessantly modifying and adapting life over eons of time. CRISPR offers the unique opportunity to intervene in the process and take charge of our own evolutionary journey. Eugenic movements have previously attempted to intervene at step 2 by determining who survives and passes their genes on to the next generation. CRISPR conceivably allows for intervention at step 1 by editing our own genetic makeup as well as that of other species. And it can be done faster and more intentionally than nature affords. Of course, many would cite the law of unintended consequences and urge restraint, if not outright prohibition, of such a proposition. But the temptation to control nature is ever present and provides an ongoing tension between that which we can do and that which we should do. This temptation was already recognized shortly after the genetic code was deciphered in the 1960s. According to Marshall Nierenberg, one of the code crackers himself, decisions concerning the application of this knowledge must ultimately be made by society, and only an informed society can make such decisions wisely. These are the questions that the current generation will have to answer, because the technology to edit the human genome has clearly arrived.